Hello.
Hi,
Hi, how are you?
I'm fine. How are you?
Okay.
Yeah, I'm just working on today. I had my lab meeting till 6:45 and it's already 7:30, so I'll just quickly wrap it up maybe. So, just to give an overview of what kind of present today. So, I went through a lot of papers which have a number of like data sets available, like publicly available data sets. So there were a lot of papers. There's still a lot that are pending that I should be going through. But I just presented like 3 or 4 papers that seem promising enough. The data sets seem promising enough. So I'll just be talking about some reading about 2 or 3 papers that we can consider as baseline and then do something about them. I don't know. I'm still not clear in my head. So that's why I've added one slide that which is titled "Help Needed from You."
Okay.
So yeah, maybe like everybody should join and then I'll just basically yeah.
Yeah, that sounds good.
Maybe.
Wait a couple more minutes.
Yeah.
And Anson, how are you?
Hi guys.
Oh, hi. Oh, sure. Sorry. How are you doing this week?
That is good. Like it was a very engaging one. I had a very tough week. I had to do some of my academic stuff. I was too much occupied with that, and hopefully I ended to the weekend. It was really engaging.
Yeah. That's.
I just have something to ask. Like, if there any issues in joining the fall semester PhD students, like in 2021 fall, there be any difficulty or anything like that by the university itself?
What was that? I didn't catch
Does
the whole
anybody
thing.
have? Sorry, Granny, I didn't hear you.
Oh, you can't hear my audio?
I'm afraid my connection is a bit poor.
Let me see if I can fix it.
Can you repeat it?
Oh, I was just listening to what you were saying. I couldn't catch all of it, though.
Okay, so some of my, my batchmates, they kind of said there could be some difficulty in, joining the next year. That is the fall 2021 batch for PhD as such. Do you think there'll be some difficulty in, in engaging people with PhD in 2021? Could there be any difficulty or something? Like they potentially won't have any funds for PhD because of which they are not going to recruit PhD students. Could there be anything like that?
Yeah, well, there could be. I know this year they might be having some problems. It really depends on, the funding that comes out of because we're still kind of in the pandemic, even though people maybe don't want to believe that. But yeah, I could see maybe potential difficulties, but it might be far enough off that they might not have difficulties. Usually they do the budgets like a year in advance. So it's probably, there probably might still be some problems. In terms of people engaging in the, programs and that, I don't know. I don't think so, but we'll see. I know this year it's going to be kind of rough because people are still, they're doing a lot of remote learning and that, so.
Yeah. Even though if they had some PhD, like it's not going to be the very same way how it has been earlier. Things, I feel.
Yeah. So you're applying for programs?
Yeah, probably in 2021 I would apply. Because I mean they didn't scare me that there would be some problem with recruiting PhD students in 2021, and I don't know what I'm gonna do about it.
Yeah. Well, I mean, it's always kind of tough to plan out a couple of years ahead, especially in this situation. But just try for the best, I guess. It's always kind of hard because PhD programs are like 5 or 6 year commitments. So they try to make it so that they can at least get you through the first couple of years if they accept you into the program. But it depends on the program. If the program's well run, then there's a better chance that things will work out better.
Okay. But some of them say that there's not going to be any issue with that, and some of them say obviously there are going to be some issues with that. I don't know what to do.
Well, I think some of them are being more honest, but I think they're also better run programs that, if there are problems, they can overcome them. So it's just, I guess it's the same advice I'd give to anyone about looking for PhD programs, is that just try to find one that's well run and that you fit into well. You think isn't going to be like a problem in terms of the administration or anything. And then, because that's another thing, if if you have problems with the way the program is run or some of the prerequisites that they make you do, or your advisor is not very good, then you're going to have problems anyways. And it's just going to be compounded by the uncertainty. Yeah. So, yeah. Well, I don't. Well, I know Jesse's not going to make it today because he's in Boston where he is visiting. And Anson, I don't know. And we were going to have Rasab, but I don't know if he's going to make it today or not. So I guess we can start by Suniti presenting on her aging work. She was going to.
Yeah. So I'll just set my screen. It's visible to all of you.
Yeah. See it.
Okay. So just quickly put together some slides. So please bear with me. So this is just basic an overview of some neurodegenerative disorders that are prevalent in the aged population generally. And so the main purpose of this PPT today, my main purpose would be just to put some ideas, just to make or explain some data sets, and then get feedbacks from all of you as to what all viable questions we can maybe explore. So I'll just start with one study that I came across. So the study talked about Alzheimer's and Parkinson's in particular. So just to give a very brief introduction of Alzheimer's and Parkinson's. So some proteins, they don't fold correctly, and then they accumulate in our brain, and which leads to such neurodegenerative disorders, Alzheimer's, Parkinson's, and so on and so forth that we'll see. So in Parkinson's we have alpha synuclein, and in Alzheimer's we have beta amyloid. Similarity between the underlying networks and the underlying expression of the proteins that are involved in both of these diseases. So they took some data set related to Alzheimer's, some data set related to Parkinson's, and identified certain proteins, the expression pattern and expression data of proteins, which were common in both. And then they came up with a human brain proteomic data set, which has, I quickly went through it. It has a lot of information about each protein, the expression data of that protein. So if we want to, let's say, find out some protein signatures related to any particular disease, or signatures which are common to both these diseases, or like maybe we can actually look at some other disease, and if the protein involved in that disease is available in this data set, then we can further maybe do something with that diseases data. So this study basically concluded with giving us a very elaborate proteomic data set of the human brain. Not CLNs, not mouse. This is a human brain proteomic data set. So they've also mentioned, they've not done anything about it, but they've also mentioned that you can look at the molecular pathways that are common for these. Or like I said, like if these proteins are common to any other disease, then we can explore the molecular pathways and then maybe find out what are the common things in these diseases and what leads to them and things like that. So they have some. They've given the data of, I think, maybe 40% population is controlled and the rest has Alzheimer's, Parkinson's, and there are some patients that have Alzheimer's and Parkinson's both. And so this was the paper that looked at it. So this data set was protein data set of the human brain. Now another one was this paper I had mentioned in our Slack group also sometime back, like way back, and this paper has first explored a lot of papers that have done machine learning in the field of Parkinson's, like machine learning on the data of Parkinson's disease, and they've compared those machine learning techniques and deep learning techniques, and then came up with like which one's better and which one's not. I couldn't go through it in detail, so I'm not mentioning anything about those techniques yet, but so the main thing that I took home was they've also learned the Parkinson's data set, which was collected like there itself. So the data set is like as well very clean and things like that. It's for 78 subjects, 55 are Parkinson's and 23 are controls. So they have imaging data in this case. MRI data and DAT scans are available for different brain regions. Right now they're giving the data pertaining to some regions of interest. So they've mentioned some numbers. But I'm sorry I forgot mentioning them. But according to regions of interest, they have taken MRI scans and DAT scans of the patients. They've done follow-ups also. So we have imaging data in this case. So related to only Parkinson's. This was another paper that I went through. Now, so this was a very interesting paper. They had gene expression data and they were predicting the age of the patients based on gene expression data. So there are these three brain regions that they targeted. Now in these three brain regions there are controls and Alzheimer's patients, and then FTLD was another disease that is like possibly associated with aging. They've said it's not claimed that it's associated with aging, but it does prevail in patients of in elderly patients. So they are looking at gene expression data of normal patients, AD patients and FTLD patients, and predicting the age of the patient depending on that. So I'm not. So this was a very lengthy, very complex paper. I didn't go through all of it, but they used machine learning in this case, of course, and a lot of statistics to predict that. And eventually they are concluding that normal patients, whatever their age is, gene expression predictions are very close to their actual age in normal patients, which do not exhibit any neurodegenerative disorder per se. And in Alzheimer's patients, even though the patients are slightly, let's say, they're 50 or 55, the model predicts that the patient is way older because they have a lot of degeneration that has happened in their brain. So this seemed like a very interesting thing that they have explored. Now they have given actually they have not given a data set, a compiled data set per se, but they've used a number of data sets and they've cited those papers as well. So like this one, this DA1, D3, they were of two, they corresponded to like two data sets that are available. Now, so these are the gene expression data sets corresponding to the three brain regions. Yeah. So right now I'm sorry if I'm very vaguely explaining what papers can be potentially looked into if you want to explore neurodegenerative disorders, specifically those papers that do report one or the other data set. So that's why I'm just being very, very vague right now. But yeah, so this one was a Harvard study. So they have they've collected data of around 60 patients themselves. Now this data is particularly special because they're following up with every patient till 60 months. Like if a first scan is taken like today, they're following the patient till 60 months and they're following up with almost all the patients. They've given some exceptions as well, like 5 or 6 patients couldn't be because of multiple reasons, but they're basically giving us, in addition to a lot of other things like clinical assessments, neurophysiological testing, and a lot of things. So the processing speed was, I mean, they gave them some tasks and then they recorded all this. Now the blood work also, and now most importantly they've also given imaging data. Imaging, the different imaging that were done were PET, structural MRI like SMRI, and then fMRI. fMRI is again like resting state fMRI, and then they were given some task and task fMRI and diffusion tensor imaging and all of that. So they have a lot of imaging data corresponding to patients, but the age group of these patients is I think above 60 age. I mentioned it? No, but most of them are above 65 age, and like 4 patients are the ones which are between 60 to 65. So, I don't know how we can use this data to maybe like formulate a question, but yeah, 65 is the age. So they have a lot of imaging data which spans over 60 months, and like each scan is done like beautifully, like this one for 60 minutes, and then this PET session ranges from 45 to, and likewise. There were a lot of task fMRI data also. Now, so these were some of the papers. I went through a lot of papers, but then they were like maybe like not cited a lot, so I thought maybe the data is not clean or maybe not trustworthy. I didn't include 4 or 5 studies that I went through, but these were the major ones that I did scan, and then like so now concludingly we do have multiple data sets that are available corresponding to, but mainly they are like for Alzheimer's, Parkinson's, or FALD in one or two cases. We have gene expression data, we have protein expression data, and we have imaging data also. Now, I still do zero in on a question that I would like to pursue even when I looked at a lot of data sets. So this is the broad interest that I would say like my broad interest. So to identify maybe so I think not this Harvard study, this study, and one even the scientific subjects. Yeah. So these had the age ranges from one of them had patients that ranged from 20 years till 75 years. So maybe I can come up with some more studies. So the broad interest would be, first, can we identify some patterns in comparatively younger brains, not in like 20 something, but maybe like in 40s or like maybe 50s. Maybe we can identify some patterns in those younger brains which can reveal if that person might develop any neurodegenerative disorder. Why I would like to pursue this question? Because A, we do know that let's say in Alzheimer's and Parkinson's, we do know that there are a lot of networks that change. There are a lot of brain regions that shrink, like in the last PPT that Bradley explained, hippocampus decreases, like the size of hippocampus decreases, and a lot of other brain regions also start to degenerate, and it's clearly visible in those scans. So maybe like maybe model or maybe explore in some other way that can we identify some patterns? Can we identify some networks or maybe degeneration in some younger brains which can reveal if that person is prone to any disorder in later stage of their life? So this would be the overall question that I would like to explore, but I think this is like very vague. So I am hoping that I'll get some inputs from all of you. And stop sharing. Yeah. Some inputs from all of you in with respect to like what all can we explore with regard to these data sets.
The Anson or Akshar, do you have any questions or comments?
I feel like I don't have enough knowledge about this to say much.
I just wanted to get some feedback from
So,
anyone.
okay, so you consider only the data that included Alzheimer and
Yeah, right now I explored only those that had neurodegenerative disorders in them right now.
Okay.
But other than that, I also looked at that I don't know my speaking. But yeah, so other than that, I also looked at the HCP dataset that was made available to us in NeuroSim. So it had fMRI time series data also for resting state, fMRI and task-based exercise. Yes, but right now I just like overviewing those papers that have neurodegenerative disorders. Addressed in
The presentation was actually interesting. I really wanted to work on some fMRI data. That looked really interesting.
Yeah, so
But I have yes.
So right now I included like only 5 papers, 4 or 5 papers that had some major data sets and the paper had some missing citation numbers. But I don't know how to like approach the question, how to zero in on a question.
Maybe this week I just try to do some literature review on it. Like I will also try to do some literature review on it. If there's any other paper that addresses this potential, I mean this question as such. Okay, maybe we'll discuss on. Even I'm actually at your position, like I don't have any insights as such, but I felt that I could try to help you with the literature and stuff.
Yeah.
Actually, yeah. So that's good. So yeah, I like the going through a couple of different types of data. I think that it's going to be hard. It's going to be a challenge to integrate like gene expression or proteomic data with the fMRI data, and that's like maybe another issue you have to think about is how do you integrate like two types of data like that, because there's a lot of interesting information in that in combining those two types of data or three types of data, but it's not straightforward. I mean, especially like, so your question is basically that you want to predict disease state from like brains that are younger. So if, what's going on in those younger brains is basically you have, there's some pattern, there's some activity pattern that you can discern. Maybe they don't use their, maybe those regions aren't activated as strongly when you're younger, and then it just leads to this pathology later. But there are probably also gene expression changes, for example, that presage this. So, there might be some, they usually measure upregulation and downregulation of different genes. So you'd have like different genes that you might be able to zero in on. And this actually would allow you, like if you went to the literature, you could actually find specific genes that may be implicated in this type of disorder. So oftentimes what they'll do in a lot of gene expression studies, or at least classically they did this, was to get a list of genes that, maybe we know from like, I mentioned gene association studies in the lecture that I gave on aging. Now, there are a lot of these different types of studies and even databases of associated genes. So you can get a list of genes beforehand and say, these are the ones we're going to look at with respect to, maybe that's predictive of something. And so you can actually maybe, take the gene expression data and find two groups, maybe one where, these things are downregulated, where these things are upregulated in a younger set of subjects, and then maybe use that to predict maybe like brain activity or something else. Like I said, it's very difficult to really kind of know what the relationships you need to look at are. It becomes pretty difficult because, of course, we're just kind of like saying, well, all these genes might be involved in this disorder, but there are individual differences and there's environmental differences, and so all these things are kind of working against prediction. But I think there's a way to do it. So, I mean, I think that's good that you have the papers picked out. And I think if you have, papers that are highly cited, that's good. It doesn't necessarily mean that the data is a lot better, but it just means that maybe people are, maybe it's a better, more complete data set, or maybe it's just that they've, have a bigger data set overall. I don't know why people are citing these papers, but usually if it's like a big consortium, they'll cite them more than not. And so that at least gives you some indication of, basically the breadth of data that you can have, if not the quality. So I think that's good. So your next steps are to like look more thoroughly at the data sets, or
I like to look at some of these data sets closely, and maybe find out if there are some common. Let's see, I'll first explore these data sets and then maybe zero in on a question eventually.
Yeah. Well, I think in this case, the question is, I think your question is pretty decent for like just kind of figuring out how you want to implement it with data. So you have, you have basically the idea you want to predict something. And then you say, well, we want to use data, secondary data. And the question is the secondary data going to be suitable for this question? And so you need to go and look at the data and say, can I answer the question with the data that I have? Sometimes it's very hard. Sometimes if it's that general, it's pretty easy to say yes, but you have to think about, like, if I want to say combine like genetic data with neuroimaging data, can I do that easily, and does it make sense to make that comparison? Because like sometimes the data sets, of course, they generate data to look at a certain question, and then you might want to reuse the data and combine it with something else and ask a slightly different question. Of course, the data isn't really set up that way. So what we've done in the past with that is sometimes we've transformed the data in different ways, like, created, principal components, or we've like taken like data and matched it up with the other data sets selectively. So you have like, you can have like a set where you have, you take your gene expression data, but it only matches up to what you have for your neuroimaging data. And it gets a little tricky in that, but I think if you can do that and create a data set that then basically answers your question, then you can move forward on it. I don't know. Also, I wanted to make sure that you knew how to get access to. Do you know how to get access to secondary data?
Yeah, some of them are available already. Like you just have to mail them and they'll like let you in. Similar to what happened in HCP dataset. But yeah, for some of them I might have to like look into how to gain access. This Harvard study they mentioned that you just have to mail us and then that'll be it. But for some other data sets I might have to look into how to gain access to them. Yeah.
Yeah, I don't know. Yeah, sometimes they have them in public repositories, and sometimes they're just kind of like, we'll send you the link. But then the other thing too is to make sure that you know what the variables mean.
Yes.
Yeah.
That is the main question. Yeah.
Well, I mean, yeah, it's and like oftentimes, you'll find data sets where they just give you some cryptic header and then like some data, and it's like, what does this mean? So we did that a little bit with the IBL mouse data. Last week we talked about the different variables.
Yeah.
So that's good to have. Like, if you're working with secondary data, it's good to kind of figure out what all the data variables are before you start working with it. And maybe I would suggest something like a code book, like what they had for the IBL mouse data, where you have like basically all the variables listed, and then how they were collected, or some information about that. And sometimes that requires you to like look in the methods of the paper and sort of figure out what that method is. Like, they'll have the data, and then you'll see a variable, and then you can match it up with something in the methods that says, that kind of mentions that variable, and then the conditions under which it was collected. And it's actually kind of important because sometimes they make assumptions about the variable or the measurement, and it's important to know what those limitations are before you dive into it.
Right. Yeah.
But no.
Yeah. Maybe then explore more and then I should look at these data sets first and maybe look at those variables and if we can go ahead with them. And yeah, I need to look into stuff deeper as of now.
Yeah. But I think that's a good start, yeah. I like that you looked at a fairly wide range of things, which I think is important if you're going to try to predict something like that. Because I think people have tried to do it, like just with fMRI data, and it's tough to get accurate predictions. Like, they're accurate if you can kind of match them up with other results, but it's not typically that easy to do a set of predictions like that. But I think if you could have like, you think about like all the data you can have for a certain study, and if you could have data that like goes beyond fMRI, it might improve your chances. But then again, it might not. So it's always it's always tricky. Well, anyways, yeah, I think that's good. A good start, and then you can update us on this. And Akshar is going to do some literature search as well, so that would be good. Okay. Well, thanks for presenting on that. That was good. Okay. So I was hoping Rasab would show up to the meeting. I haven't heard from him this week, so I don't know. but he was planning on he wants to start working with the lab. He's planning on doing some things, but I don't know what they are exactly. He's probably still trying to figure that out. So maybe next week he'll present on what he want. Like asked him to do like a medium length sort of plan for what he wants to do, and just interacting with people. He's been on Slack if you've seen a couple messages from him, so that's a good place to interact too. I forgot to ask, did you want to start a Slack channel for this aging topic?
We started it already.
Oh, you did?
You did make one, yeah.
Okay,
You
good.
did make one, it's there.
Yeah, I think I remember now. Yeah.
Yeah.
Okay. Well, that's good. So, let me see. Akshar or Anson, do you have anything to add before we move on, or did you want to mention any news or any items that you want to bring up today?
Actually, not with the aging thing, but yeah, I couldn't hear you properly in between.
I can't hear you.
Okay, so
Okay.
I actually looked into the the net logo thing, and you guys can hear me.
Yes.
Yeah.
Okay. So I was trying to look into the net logo thing earlier this week, and then I actually got up with my work. I actually was trying to do stuff like checking out some important points to be assessed in case of glaucoma. It's how I was thinking about it. I thought I could present about my idea sometime, but not this month, by next month or something. I just thought I could have an idea about it and present it to you guys, maybe.
Yeah, that would be good. I can put you on the schedule when you're ready. You can send me some slides beforehand, or let me know what week would be best. And then I'll put you on the schedule, and then we can go through. Something like Suniti's talk would be fine in terms of length. Maybe 20 minutes. And then, we can talk about, it doesn't have to be super polished. It can just be like, here are my ideas that I have. This is, kind of where I've gotten so far. And, maybe pose some questions for, continuing on with what you're doing. It should be enough for some feedback. Maybe if you're like creating, actually, I don't know how far you'll get by then, but we'll see. But yeah, that would be great if you could do that next month sometime. Good. Anson.
So I've been mostly preparing for the fall semester, reviewing Python, calculus, and done preparing for my fall courses. I'm a little booked right now with that preparation, but I'm thinking maybe some ways I can perhaps fare on some of the things I'm reviewing, maybe, and then once the semester starts, I might have a little more time for the lab. The semester starts in 2 weeks, so I'm doing some last minute review before the semester starts.
Yeah, that sounds good. Are you having in-person classes, or are you having like online classes?
So 2 of them are online and 2 of them are in person. But I think the in-person ones have reduced hours to only meeting once a week. And I'm taking a big data with Python course, a artificial intelligence, and a computer science capstone course. I'm not a computer science major, but my data science programs ran by both the stats and CX departments at my school. And the fourth one, some other data science upper level course I forgot, to be honest. But yeah, I just wanted to make sure I'm organized and have be the prereqs before the semester starts, so I can jump in with prepared.
Sounds good. Yeah. Definitely. So yeah, looking forward to fall semester and next month and everything. And
yeah.
So I'm gonna present on some things now. Let's see. We'll do the entire screen. All right. So first thing I wanted to talk about was, and I know some of you have been. I sent this out in Slack, I believe. And this is the critical periods in development Discord. And so this is a new chat. This is kind of like Slack, but we're running it for specific topics. This critical periods Discord came out of the Artificial Life Conference. So when I went to the Artificial Life Conference, we talked about our presentation there, and then we had a discussion afterwards. And there were a couple people in the group who wanted to form a discussion group, Jason Yoder and Kazia Kazdan. And they're going to be in this group. And then I see that Akshara and Suniti joined. And then Jesse joined as well. So welcome to the Discord group. And Discord works a lot like Slack. You just post a message in here. You can use emojis. You can post files. So we have a couple of readings here already. We have one on modeling the evolution of sensitive periods, and then 2 papers on critical periods. And if you're asking what critical periods are, I'm about to give a presentation on that, so that will be answered soon.
And just because Bradley mentioned my name on Discord, I appeared here in Gisty as well, for whatever reason.
Okay.
Hi.
Hi.
Surprise.
Okay, yeah. Well, good. Thanks for showing up. So yeah, this is gonna be like a discussion over time. People will read papers and we'll post things here, and we'll see where it goes. So that's this Discord. Then I wanted to quickly remind people that we still have our issues on GitHub, and we have the going back to the Neuromatch Academy. So we still have this complexity of feedback loops project that we're talking about. Not this week so much, but going into the fall, we'll be talking more about that. We still have this epistemological directory, so this may be more for Jesse, but I don't know if we want to continue to work on this, but we have a pretty decent set of topics in here, and so we might want to get other people involved. If people want to contribute to a topic, that's something that you can do. If you're not in the meeting right now, this is especially true for you. If you want to contribute to this, this is a resource for people who might be interested students in NeuroMatch or in even outside of that, but they're interested in neuroscience more generally. So we have a number of topics here that we kind of worked on putting in intro material for just general neuroscience, or putting in like philosophy resources. And so that's something that is something if you contribute to, you get your name involved in the in this release, and then other people can learn from you, and you get some, you might become an expert in a topic or something. There's also Jesse, I think, mentioned the Slow Neuromatch Academy, which is a version of the Neuromatch Academy where people are going to be walking through slowly the materials as opposed to the way it was presented at the actual Neuromatch summer school, which was very fast. And so that might be something that people want to go back to those references for in a bit a slower fashion. So I think we talked about this in Slack, Jesse and I. where these are materials that probably you can use for years to come, not just in a 3-week summer course, but they're open materials, and to unpack the materials in a way that helps you learn or helps other people learn. I think that's useful. We might actually, Jesse, add a slow neuromatch tab into this directory. I don't know if you want to do that, but we can do that later.
Yeah, I know there was a slow. I think there might be one added. I don't know. Was it one added somewhere else or mentioned that? And just to say a bit more about that, hi everybody. I'm a bit late. I'm semi on vacation, traveling and whatnot. But for the slow neuromatch stuff, basically what's happening is there's different collections of people who are on their own. It's not an organized thing for neuromatch at all. And it's sort of like these sort of people with different making their own groups or pods, if you will, to go through the material again at a slower pace. I'm going to try to host one, probably maybe a week or two at this point. After things, I have a lot of things that I'm doing, but I think a lot of people are in the process of doing that, going through material again, and then kind of adding their own spin on it. Like I have my own interest that I'm going to add as a bonus material. And I think for anyone who's interested in the work of the lab and watching this, like there's a lot of either welcome to make their own or join mine, or even if they want to sort of do their own thing and then meet up to talk about it in sort of like a super pod sense or in a sense in relation to the lab itself. I think that would be very useful. So, there's a lot of good things happening there, and however we can, whatever the best way to incorporate that into our existing materials in our GitHub, and definitely the materials that NeuroMatch created will last, for some time, and we're intending to go back to. So there's going to be a lot of remixes on them. I think that's exactly what they were made for.
Yeah, that's good. That sounds good. And so moving on from that, if you have any questions, we can talk about it in Slack. But I just wanted to bring that up into people's consciousness again. So let's see. This is a paper. I don't think we're going to have time for it, but I can actually talk about it really quick, and then maybe we can talk about it more in the future. So this is something Jesse was interested in. That I actually found on in a literature search independently of him, but this is called Scale-Free Biology, Integrating Evolutionary and Developmental Thinking. And this is a new paper in Bioessays. And this is by Chris Fields, who's an independent researcher, and he's done some really good work in philosophy of science. And Michael Levin, who's done a lot of work in the electrophysiology of cells, not just neurons, but other cells as well, stem cells and other types of somatic cells. And he does a lot of really interesting work. So they've collaborated on this paper. The abstract is, when the history of life on Earth is viewed as a history of cell division, all of life becomes a single cell lineage. The growth and differentiation of this lineage is reciprocal interaction with its environment. In reciprocal interaction with its environment can be viewed as a developmental process. Hence, the evolution of life on Earth can also be seen as the development of life on Earth. Here, in reviewing this field, some potentially fruitful research directions suggested by this change in perspective are highlighted. Variation and selection become, for example, bidirectional information flows between scales, while cooperation and competition become scale relative. The language of communication, inference, and information processing become more useful than the language of causation to describe the interactions of both homogeneous and heterogeneous living systems. Emerging scale-free theoretical frameworks such as predictive coding and active inference provide conceptual tools for reconceptualizing biology. And they talk about this. Like a lot of ideas that we've talked about in this group, but some that we haven't. We've talked a lot about things like cooperation and competition and predictive coding and active inference. But we haven't talked too much about evolutionary stuff and developmental stuff. So we haven't really talked about cell lineages too much, or history of life, or cell division. So what they're doing is they're bringing together a lot of biology, especially like developmental biology and evolutionary biology, but they're bringing that together with maybe like neuroscience and some complex systems theory. And I've read it a little bit. I haven't really made a deep dive into this paper, and this is something that Jesse said he might be interested in. But it has some interesting insights, at least. And so they start off with the history of life. And so the history of life is phylogenetic, meaning that you start with a single common ancestor and you move outward over time. And over time you get more and more taxa or species. And that gives you the diversity of life you see on Earth today. And of course, we have to characterize this process somehow. Most people have used what they call a tree, but biologically that doesn't mean a lot. It isn't like a biological tree, it's just like a logical tree. And so they kind of bring together this idea of tree thinking with development, which is also, in a way, tree thinking. And they make the point that there's a connection between development and evolution. They also talk about scale-free methods, which are ways that you can measure systems independent of their scale. So, it's like measuring things in the aggregate. And there are mathematical tools that allow you to do this. They actually talk here about the free energy principle, which is, of course, something that everyone talks about these days, but I guess they're going to throw that in and contemplate it a bit. Like I said, they're mixing in like evolutionary and developmental biology with sort of cognitive neuroscience or neuroscientific methods that they talk about. So if you're interested in these topics, it's a good paper to read. They kind of get into this notion of evolution as sort of like error prediction or predictive processing, which I think is interesting. I'm not really sure if that's like something that would be widely accepted, but it's something to think about. Definitely people have tried to combine like cognition and evolution before, but I think this is a nice take on it. It's certainly there's certainly a lot in this paper. So are there any other figures here? So they just advocate for an integrated scale-free biology, which isn't really new, but they're kind of working on, kind of stitching together the sort of the intellectual territory here. So they say, what will this integrated biology look like? As outlined above, we expect it to render concepts such as randomness, goal-directedness, competition, cooperation, causation, and communication scale relative. It will consider both organisms and superorganisms, supraorganismic structures like beehives and ant hills and things like that as environments for systems at smaller scales, as is already happening on a smaller scale in cancer biology. So in cancer biology, they tend to look at cancer as being like the product of different scales. You have cells, you have tumors, and you have the body. And, that's how people were modeling that. And so they're kind of advocating for that type of approach, but for all of biology. Again, like I said, this isn't like absolutely novel, but they do a good job of pulling this together. So that's my 5 minutes on that paper, and I can give you the link to the folder in case you want to look at it.
It seems directly related to my research interest, and I'd definitely like to read that.
Yeah, there's the link to the folder. And, it's probably a bit dense, so we'll probably talk about it more in future meetings.
Yeah. And similar to Anson, I mean, I don't know how much I've really mentioned it here recently, but some of my interests are I'm looking at different ways of organizing certain educational ways of thinking about different fields of study. I've talked a lot about like cognitive science, but I think I think this is directly related to that effort. So I'm slowly making some headway in related projects to that, and that seems like a good paper that would fit with that effort. So anybody's interested in that, feel free to talk to me about those things in Slack or so on. But, yeah, thank you for sharing that here, Brad. And talk about it later too.
Yeah. So, and then finally, I'm going to get into this critical periods. This is just, I wanted to go over critical periods a bit just to introduce it to the group. And this will be probably useful for people more generally. But so I've been looking at this. I have a bunch of talks that I've done over the summer, and I think we might have to end up putting them into a collection or something because there are about 10 of them now. So we can talk about that in Slack. Maybe it'll be good for like an educational curriculum or something to go through these different things. But so these are critical. So we're calling this critical periods in developmental learning, but you'll find that this is also sort of a biological concept that stands alone from developmental learning. And this is a tweet that was posted this week. This is like a question that someone posed. What is the one capacity that best differentiates biological learning from machine learning? And there were a lot of interesting answers to this. But this is one of the things that I think we can say is that biological learning has critical periods, which is not necessarily true for machine learning, at least not right now. And so critical periods are not only important for the initial acquisition behavior, but for physiological regulation, and you'll see that. So I wanted to point this paper out to people. This is a deep learning paper from last year, and this is kind of how they're approaching development in deep learning networks. So we don't think too much about development in terms of learning and deep learning and artificial learning, but we do think about learning in terms of structure and in terms of especially linguistics. There have been some great linguistic advances in simulating linguistic reasoning and things like that. GPT-3, GPT-4, and the other, or GPT-3, I think is just the one that they have now. But this is basically a take on development in deep learning. So they're using words, items, and properties, and they're looking at how the network acquires these over time. So this is developmental time. These are items that the network learns. These are the properties of this item, and this is developmental time. So it's learning items, and then it's learning its properties over time. And it can learn for different, for the same item or for different items. So it can have multiple attributes for one item, but other items can acquire properties as well. And then this is an example of how the network sort of relates these things. It can actually predict items by their properties. And so you have these items and then the properties, and you have an actual thing, you want to predict it, and it's able to do that. And so they talk about this as semantic development. But this isn't really development in the way that biology does it. This is a way that they've done it in deep learning networks. And I think there's a big opportunity for looking at development in like artificial intelligence and deep learning, because people really haven't approached the topic in a very sophisticated manner. So sensitive periods themselves are defined as periods of development that enable periods of high information acquisition. So I used the word period twice there, but basically this is the developmental time as it unfolds. And these peaks here, these distributions are sensitive periods. And so the sensitive period is a window in development that enables these periods of high information acquisition. So in this case, you have this axis is plasticity. So this is the sensitive period allows for plasticity for these different functions. And so as we can see here, this is an example from this organism, and it's the V1 area of this organism. So this is where the eye is opening up in development, and you have this first peak of direction selectivity. So there's a critical period for direction selectivity to be acquired in the organism. If you, right after the eye opens, this is when the greatest amount of learning occurs for direction selectivity, for tuning the cells and things like that. But then you can see that it shuts down after this early period, and it doesn't allow for additional acquisition to happen, or at least as great as it would during this critical period. The same thing happens for ocular dominance. This happens a little bit later. This critical period opens up and then it closes. And so this is when most of your ocular dominance is trained into your V1 area during this period of development. And then slow wave sleep has a wider critical period. But again, like the others, it shuts down after a while. And then you don't have that ability to learn or to acquire information at such a high rate. And so one of the ways in this paper by Takao Hench, which is on the Discord, he talks about the specialized molecular milieu for plasticity. And so what that means is that there are molecular events like gene expression and proteomic activity and other things, and they influence the formation of dendritic spines and axonal rearrangements. And so this is an anatomical phenomenon. This is a physiological phenomenon that is enabled by this during this critical period, and it allows for this enhanced learning over these short periods of time. And so basically, it's a reorganization of the anatomy, and so that leads to this sort of ability to acquire information at a high rate. So then how are critical periods regulated? We talk about them being like opened and closed. How do you regulate them? So what happens in a critical period is you have this inhibitory and excitatory balance this is in the brain amongst neurons. You have this inhibitory and excitatory balance amongst the neurons in this network. And so as I understand it, there's this balance. You must think of it as homeostasis, where the network balances, and then eventually it becomes out of balance, or it becomes balanced. I'm not really sure which, but it basically triggers this critical period. As you can imagine, in early development, you're getting a lot of connections formed, and you're getting a lot of cells put into place. So you're getting this balance of activity, and the critical period then is where you have to regain this balance, or you have to enhance it in the face of a lot of this reorganization that's going on. So there are signals that happen in that part of the brain, or wherever the critical period is linked to, that enable this acquisition and reformation. And then there's some molecular break that happens at the end to end the critical period. So the critical period is shut down with this molecular break. It usually, again, has to do with this balance between inhibition and excitation, and then that's the end of your critical period. Now, learning can still occur after the critical period, but this critical period is really this period of great plasticity. And in this work, Takeizian and Hench, they talk about this occurring across brain development, and they give a lot of good examples of how this critical period is linked to stability in the brain and brain activity. And so there's a lot more information in that paper. One example of a critical period is in the formation of binocular rivalry. So binocular rivalry is a pretty famous result in neuroscience. Hubel and Wiesel did this in kittens. So they looked at binocular deprivation. So binocular rivalry is where you have your V1 area and you have two eyes, and your eyes are just opening up, so they've never seen the environment before, and your eyes have to they compete for connections within the brain, the right field and the left field. And during binocular rivalry, these two eyes, they compete for connections, and it isn't given that they're going to balance each other out and serve as like the way we have our eyes today, where you can see this red and this blue text, and you see it as overlapping. But it's because you have binocular vision. If you wear a pair of these glasses, they polarize the light differentially in each eye, so you can actually make out either the red or the blue depending on which eye you're looking through. And so our eyes are can distinguish like depth and other types of cues, and they cooperate. But that's not a given our developmental. If you take one eye out early on in development, you end up with a far different visual system. And so there's this need for plasticity in development to develop binocular vision. And this critical period is actually pretty essential in determining how this is structured. So you can look at this neurobases of binocular rivalry and then this original paper and get a sense of how that works. Basically, it works through a series of, basically sculpting these circuits in the brain. So you have this feedback, you have this set of feedback connections where you have one column devoted to the left eye, one column devoted to the right eye, and you have these different mechanisms, top-down feedback excitation, and some other types of connections. You have lateral excitatory connections, so you have connections that go between columns, and then you have inhibitory connections, which also sculpt this. And all of these are really influenced through the critical period. The critical period basically determines how this is all structured. And so if the critical period is widened, it might, in say like evolution, it might allow for these processes to be optimized more or continue or whatever.
Can I make a slight note for new people watching or whatever, or those who aren't here meeting? I just say, if people aren't familiar with our work in brain brake vehicles or developmental brain brake vehicles, it's a really cool thing to note that if you look at Valentino Brainberg's book on vehicles, if you're not familiar with the book, he kind of talks about, he goes through these very simple thought experiments and builds on each one in a sort of toy example of almost as if it was an evolutionary view on the development of certain nervous systems or whatever. And he does that in a pure thought experiment when the second half of the book, the later half of the book, or sort of like the appendix, however you want to say, like he goes into detail about this. And one of the main things is actually this, about having crossed wires and how your eyes interpret information and why is there different, how the eyes develop and the wiring of them and things go together. I don't think it goes into this particular aspect of it very much. But I just wanted to mention that it's actually a really cool thing and actually related to a lot of the work that we've done in the past. Even though I don't think it's quite been. I'm not saying, this is exactly what we did before, not at all. But more like there are actually a lot of ties into the things that we've been looking at in the lab that fit really strongly to this. And if people are new or are curious about them, that'd be a great thing to talk about in Slack, because there's a lot of interesting, maybe not direct, but like 1-step, 2-step connections to past work here, and I think it's a cool thing to think about.
Yeah, when I was putting this together, I didn't think about putting the Bradenberg vehicles in, but I think this is actually a good example of kind of like how, like our Bradenberg vehicles were trying to add connections in and neurons in and to show like you can develop something that's you can develop a vehicle nervous system and get different, maybe more complex behaviors than what Breitenberg got out of them. So this is an example again of like the complexity of this binocular rivalry and how you have a critical period. You have this period where there's great plasticity and all these different types of connections, as opposed to having everything totally connected together in a big mass, or having things connected, one step at a time. So you'd have these really inefficient circuits. This critical period allows you this flexibility to rewire quickly and get something that's pretty optimal. And so you could do the same thing in the vehicles as well. You could have a critical period where there's a lot of rewiring, and then shut down that critical period when you hit maybe like we were doing things with genetic algorithms, and so in genetic algorithms you have something similar to that where you keep evolving your network in this case, and at some point you hit like an optimum, and then you can very well evolve away from that optimum if you let it evolve too long. So you want to protect, sort of the optimal solution that you've gotten, so you can actually sort of modularize the problem, where you evolve different modules at different times and then stop evolving it when you get to something that looks reasonably like an optimum. And so those are all like ways to approach critical periods. But I think, I think if you combine critical periods with Breitenberg vehicles, we could really, it would really be interesting. It'd be interesting. So this critical period activity shuts down after a while, and you end up with what you have in adulthood, which is this ability to have binocular vision, and it's fairly efficient. But then you have other types of critical periods, and these are more involved with the physiology of the organism, not necessarily the brain, but in terms of growth and in terms of just the timing of development. So I wanted to present some things on that to give people an idea of developmental timing as a thing that is really at play here. It's not really just about the brain and its connectivity. It's really about timing and, turning things on at a certain time in development. So this is diapause as a developmental delay. And diapause is a term that is used to represent this sort of freezing of development. So in a lot of invertebrates species, you have this diapause period where there's some environmental signal like drought or starvation, and it will basically suspend development for a while. And then the organism will pick up after it comes out of diapause and continue to develop. So this is like a flip side of the binocular rivalry idea that we were just talking about, where you have a lot of dynamic reorganization. In this case, you're freezing the phenotype and you're allowing it to survive without too much impact. And then when the stress goes away, you can pick up development where you left off. So this is diapause, delaying the developmental clock in response to a changing environment, this review down here. And they give a couple of examples from different invertebrates, actually this is not an invertebrate, but this is a killfish. This is Austrofungulus limonaeus. This is in the embryo, where you have this diapause and this escape from diapause. So you have this, really early in development mechanism where it can go into diapause and then the egg can escape diapause. You have this hawkmouth, which is insect or an invertebrate, where you have this. This is controlled by hormones, so these letters are hormones, and hormonal levels trigger diapause, and will also allow the organism to escape diapause. So in the pupal, in a pupation period, the larva is able to trigger this pupation period through different concentrations of hormones. Then there's this block on the hormones, and that allows the organism to go into metamorphosis. And then there's some temperature signal, and then it can stop metamorphosis, and then it can resume metamorphosis depending on environmental signals. And so this is all like, pretty, I think it's controlled by genetics, but it's also controlled by hormones as well. And you can see this in C. elegans in the Dower period. So in C. elegans, you have this egg, and it hatches, and then you have the series of these larval stages from L1 to L4. And then you have the adult. So these different larval stages actually, L1 and L3 actually starts in L2, but in L1 you can have a starvation response where basically if you starve the worm right as it hatches and into L1, there's a starvation response where it can survive for long periods without food. And it retains its adult growth with maybe some consequence to it, but it doesn't die. So it's put in this low metabolic state. And then from L2 to L4, there's this alternate pathway. If there's a very limited food supply, or if there's some other pheromone signal from other worms, like there's crowding amongst the worms in a certain environment, you get this Dower stage, which is this sort of stage where the worm grows this thick cuticle, and it's very, lethargic, and it's conserving energy. And so you have this diapause stage that again is this sort of critical period where, if you didn't have this response, there would be different environmental fluctuations that might kill off the organism before it reached adulthood. But these diapause stages actually allow it to survive unfavorable environments. And so you have this, and this acts in a similar way. There's this critical period window where it's acting at a certain time in development. It enters this period, and then something happens. Either there's a lot of activity, or there's it basically shuts down, and then the organism can recover from this, and it's programmed so that, maybe there are environmental cues that trigger the recovery, or timing cues that trigger the recovery, or some molecular cascade that triggers the recovery. And so then this all has some sort of fitness consequence. And again, one of the reasons why you have this critical period is because it's a protective mechanism for the organism at different points in critical points in development. I don't fully understand why it happens, but it probably does relate to fitness. And so this is another example. These are hormonal profiles from 3 species, and this is where they go through this embryonic diapause. So this is a little bit different form of diapause. This is embryonic diapause, where you have basically this suspension of sort of normal physiological activity during this diapause window, and then a reactivation afterwards. And so, you have hormonal fluctuations during the entry of this diapause. It's usually due to something like food fluctuations, or temperature, or photoperiod instability. And so in the mother, you have this response, and diapause is marked by hormonal stability, and then the concentrations change markedly after reactivation. Basically, this freezing of this hormonal stability is related to just this, it's a protective mechanism to the embryo, so that it doesn't get impacted by these fluctuations in resources and things like that. But this also relates to something more theoretical, and it is this idea of developmental delays. And so I do preprint out on developmental delays. And so the idea is that you have these 2 growth trajectories, this blue and this red. And the red growth trajectory is always compared to the blue growth trajectory, so that you have this comparison of different growth trajectories and their differential. In this top example, you have one where they start out at the same point, but this red one grows more slowly than the blue one. So the growth is decelerated. And this could be due to a number of things. Could be due to environmental factors that are, as nice as they are for the blue one. But there's this lag over time in growth. You can also have lag at the beginning of development. So in this case, you have the blue trajectory and the red trajectory. The red trajectory is the way at the beginning of development and then continues along the same trajectory as the blue trajectory. But it ends up in roughly the same place as this top example. And so there are different ways you can play around with this. And this is an example of C. elegans, or the egg, hatching into the adult. And so the idea would be that you would have different eggs that would hatch, and then they'd grow to be different sizes. That's what this represents. And we can actually model this using delay differential equations. So we can characterize both the absolute changes and relative rates of change in development. Now, when you add critical periods into the mix, you actually end up with a much more complicated function here. So you actually end up with maybe like enhanced growth over a certain period of developmental time, or a decelerated period of growth over that part of developmental time. And it usually has to do with something internal to the organism, something that the organism needs to develop. It could be like visual acuity. It could be something like survival of the embryo or survival of the larvae. So there are a lot of reasons why you would have this, but you can map them out in this way to kind of show how this works. Now, in these examples, we don't have like critical periods. This is just like uniform development. But if you read this preprint, I talk a little bit more about developmental incongruity, where you have these differential periods in development and how those change, and sort of how we can think about that theoretically. So that was a pretty broad overview, but that's something. I'm trying to not share my screen anymore. So that's something we can talk about more. But do you have any thoughts about that? Or?
Yeah, it's interesting for me because actually, my first actual, well, when I was in a neuroscience lab in undergrad, we were looking at the circadian rhythm and whether or not it was present in mice as they were developing from very early stage. And that was sort of like our experiments didn't go well for a number of reasons. We didn't really get any results, from what I understand. It was part of a broader look at the circadian rhythm, but it's definitely something that I'm interested in terms of how it would affect learning. And I don't really think about the MBA. I didn't. If I saw the original tweet that you mentioned that asked the question was, I don't know if I saw that or absorbed it. But I mean, I think that's a really interesting thing to look at in terms of what are the differences there, and these differences in particular between that and even the previous paper about scale and other things are all kind of very interesting vein. Do we talk, since I wasn't here earlier, do we talk about the neuroscience and reinforcement learning paper?
No, we didn't.
Okay, well, it's not necessary, but just saying, there's a lot of interesting energy that's come up this week in the lab about these sort of takes on things. So I'm interested in where they go for sure, and it's really interesting to think about that in terms of. I don't really have it on top of my head right now, but the previous work about circadian rhythm, because I think one of the interesting ways to look at the difference between machine and biological intelligence in that sense is that actually this came up with a question. If anybody, I don't think anybody else was there at the MIT BMN summer school course, but there was a question: how would machine intelligence be able to digitize or quantify like the kind of communication information that hormones and chemicals would possess in terms of how it would affect development of things, or at least affect at least in a way that wouldn't be, compared to straight machine learning as it is now, which is more just like straight information. And it didn't go into this. It didn't at all connect the dots all the way to those influences on biological learning in different stages. But I feel like it's a very interesting. Okay, here are a lot of unknowns, and where could they go? And that this paper is pointing at them. And I guess this is the kind of thing that we talked about more in Discord about this topic.
Yeah, well, yeah, you might bring it up on the Discord as well. I mean, I just kind of glanced over the stuff about digitizing hormones. Yeah, that's a good question, because I know that people have been talking about taking neural networks and making them more realistic, which means like adding in, molecular aspects to them and signaling aspects. So, hormones and gene expression and cellular signaling and things like that. But they have to be, they have to sort of figure out how to represent that stuff, and then, in a way that's not just kind of a big mess. Something that, because the reason why we have neural networks as they stand is because people, have, there's this idea of abstraction where you want to abstract the most important elements of some process. So, people thought, of course, well, connections between neurons, the electrical connections are the most important things. And of course those work, but, there are other things going on in the brain as well. So.
Yes. Yeah, there's a lot of anything to say about that, but I'll leave it at that for now.
Okay. Any other comments or questions?
I know we don't talk about it, but did other people present earlier today? I'm sure that was well. I'm sorry I wasn't here for that, but I'll definitely check the recording of it.
Oh, yeah.
And look at other presentations earlier today.
Yeah, Suniti presented on her project Aging, so she's investigating a couple of data sets. So.
Okay. Very.
Yeah. Next week we'll probably follow up on that, and then we'll probably maybe Rasab will do his presentation on just it could be a discussion. Doesn't have to be a full-blown presentation on his interests, and then we could talk. Akshara maybe wants to talk about something, or Anson. I mean, it's just something. If you find something interesting over the course of the week, it doesn't have to be a lot of time involved. And then Jesse, I don't know if you're planning on something for next week.
I mean, I'm kind of going to be. I may be less active the next week or two, but I think that even on my vacations, I show up and have a lot to say because it's the kind of person that I am currently. So, yeah, there's a number of things for me to talk about that I really have to spend some time to kind of crystallize a bit more. But I mean, there's a NeuroMatch slow pod. There's the cognition study group. There's some efforts to create more educational materials that I've talked about. And there's also, I don't know if you knew earlier, but there's NeuroMatch, Super NeuroMatch, which is the conference that's, essentially Society for Neuroscience is the big conference in neuroscience, and it's not happening. So I kind of feel like, Super NeuroMatch is sort of like, okay, Avengers assemble once again, and the low organizers of the NeuroMatch are kind of putting on a big 5-day conference that's going to be very encompassing for a lot of neuroscience stuff. And everybody here should probably be looking into that or whatever. But I mentioned to Brad, like, hey, why don't some of the topics we mentioned today? I was like, it'd be interesting to talk about. Do some work for this in preparation for NeuroMatch. I think it's happening in October.
Yeah.
I don't remember when.
End of October, I think.
So I mean, there's some time, and that's kind of a nice goal to work towards for things. And obviously, there's the existing NeuroMatch feedback complexity project, which is still in the docket, and I'm mentioning that because it still is for me. But I mean, there's a lot of cool stuff to be discussed. So for anybody who's interested in any of those topics or other topics, please mention it on Slack, and we can build out stuff for anything that anybody's brought up or is interested in. So.
Okay. Well, thanks. If there is no other comments, thanks for attending. I hope you have a good week, and hope everything is stay on top of everything. I know people are getting back to school, and but if you have any comments or questions, you can ask them in Slack. Okay, everyone. Take care.
Okay. Take care. See you next week.
Bye.